Good morning, everyone. I'm Susanna Perencheska, Program Director at the Division of Neuroscience at the National Institute on Aging, and it is my pleasure to welcome all of you this morning. The ADD Drug Discovery Conference is, as Howard mentioned yesterday, is supported by a conference grant by the National Institute of Aging for the last 5 years in collaboration with the National Institute of Neurological Disorders and Stroke and Office of Rare Disease Research. This conference for our institute is a very important educational vehicle. We support a number of funding initiatives for translational research, and we would like our customers to be well equipped to be successful when applying to these funding opportunities. So it is wonderful to see how this conference has evolved over the last 5 years, how it provides not only a crash course for drug discovery for the biologists, but also tackles the hot issues in the science of drug discovery for neurodegeneration. So it is always terrific to see all the young talent and the drug discovery wisdom in the same room, the biologists, the drug discoverers, the pharmacologists. And above all, it's a great network opportunity for all of us. So without further ado, it is my pleasure to introduce the chair for this morning's session, What's Druggable? Designing Drugs for CNS Target Classes, Dr. Mark Frazier from the Michael J. Fox Foundation.
Okay, thank you, Susanna. And I'm Mark Frazier. I'm Vice President of Research Programs at the Michael J. Fox Foundation, and we're very proud to be a sponsor of this program, and I want to congratulate ADDF staff for a wonderful meeting. There were some excellent discussions yesterday, and hopefully we can continue them today. I'm excited to be chairing this session on CNS drug target classes, and there was a lot of discussion yesterday about single molecular targets versus pathways, and I think we'll continue that discussion. But the Fox Foundation has specific funding mechanisms for studying targets within Parkinson's disease and validating targets within Parkinson's disease. So I just wanted to plug a little bit about what the Fox Foundation supports and what we have in our pipeline before getting into the goals of the session. So we feel very optimistic about what's in the Parkinson's pipeline. I've listed a couple of targets that we are currently supporting, both for symptomatic and disease-modifying approaches to Parkinson's disease. We also fund from target validation to more drug discovery, drug development programs. We have over 100 industry-led projects that are moving to the clinic. We're focused again both on symptomatic treatment for unmet symptoms of Parkinson's disease, untreated symptoms of Parkinson's disease, and disease-modifying therapies. We've also developed a number of translational research tools that I just want to share with you very briefly for those that may not be familiar with them. I think they're valuable tools that may help accelerate your research. But we also are supporting over 20 clinical trials, and these are interventional trials focused on things such as trophic factors, an alpha-synuclein-based approach, and some mGluR5 programs that are being developed for treating dyskinesias. We've also recently launched a repurposing program. This is focused on drugs that have been developed for other indications that may have applicability to Parkinson's disease. So I encourage you to look at our website and check out those programs as well. So very briefly, I don't want to go into too much detail, but just share with you some of the tools and resources that the Fox Foundation has developed over the last several years. We've funded a number of transgenic rodent models, knockout rats, genetically engineered mice, and funded the characterization of these animals in a standardized way in one lab. And the data on these animals is available in real time as we collect it on our website. And so if you do not currently subscribe to our email list, please email the research inbox at the Michael J. Fox Foundation and get on our list, and you'll receive these updates new animal models as they are developed. We spent a lot of time and effort in reagent develop. Our first foray was into antibodies against the LRRK2 protein, but since then we've developed a number of assays that are commercially available, as well as viral vectors that will be coming down the pike very shortly. We've invested a lot in brain and body donation programs. We have tissue available that we make available through an RFA mechanism through a program in Arizona. And then there are a number of studies that we've supported at various levels collecting biologic samples from both people with Parkinson's disease and controls. This could be serum, cerebrospinal fluid, urine, etc. And so I encourage you to check out our website to understand what's available. And there are funding mechanisms associated with these resources if you'd like to use them. And then finally, we have invested a lot in developing clinical research tools, so scale validation to understand what is the most appropriate scale, for example, to measure dyskinesia or cognition within the context of Parkinson's disease, so that when we do have drugs that are ready to be tested, we have the appropriate ways to actually measure their success. So, as I mentioned in the beginning, this session is focused on CNS drug targets, and I've seen many iterations of this slide from different companies and different organizations, but it's an attempt to, and this was, I have to say, adapted from several different organization slides, but it's an attempt to create some sort of matrix on evaluating a putative molecular target, and I think this can be applied to, again, a single molecular target or potentially a target pathway. But it's a way when the Fox Foundation undergoes our prioritization of molecular targets or pathways, we try to understand what's known and what's unknown about these targets. And so starting from the top, we try to understand what is the expression and activity of this particular target, from broad expression to a narrow expression in the disease organ. What's the preclinical pharmacological target modulation data? Has there been modulation in vitro? Has there been ex vivo modulation, et cetera? Or has it gone to in vivo modulation and actually shown a beneficial effect in Parkinson's model? And so, and then further down, we talk about the non-human genetic modulation. So, are there any knockout or overexpressing models that we can learn about this target from? Are there any human genetic studies associated with this particular target or pathway? And what is the strength of the data around those genetic studies? And then we go into the clinical target modulation. Has this target been shown to be modulated, inhibited, activated, et cetera, in the clinic? And this refers to non-Parkinson's disease indication, and we kind of can sort of grade the result of the clinical studies, whether it's a negative result or all the way to effective and safe and pivotal trials. And then finally, we get to the clinical target modulation in Parkinson's disease. And again, we sort of rank order the data on where that validation is. So I don't know that we have to necessarily check all of the boxes when studying a target, but it's a way to, again, try to understand if we are going to validate a Parkinson's target, what data could be available, what data is available, and what data actually should be available before moving into the clinic. So without further ado, I want to move into the speakers. I'm very excited for the speaker list. First, we'll have Gerard Rosset from Dart Neuroscience, who is going to be talking about protein-protein interactions as a molecular target. Ravi Kurumbil will then talk about kinases, and this is particularly relevant to Parkinson's disease, but as well as Alzheimer's disease. There are a number of putative molecular kinases that are molecular targets for Parkinson's disease. Sean Stauffer from Vanderbilt will then talk about GPCRs and ion channels and things you should consider when evaluating the druggability of these targets. And finally, Guruk Basi from Elan will be talking about biologics as targets and some of the lessons learned at Elan. So just one final comment. It's kind of interesting being an observer on many different review panels of when evaluating targets, it's one thing I've learned is that it's clear that different individuals, investigators, organizations have different risk tolerances for what target to prosecute. And so I would just make the comment that there are targets out there that some people think that are just untouchable. Some think that are actually very tractable if we just have the right tools. So I'd like you to keep that in mind throughout the talks, because I think it applies to many of the targets that will be discussed today. So without further ado, I'd like to introduce Gerard Verset from DART.
Thank you, Mark, for the very nice introduction, and I'd like also to thank Howard Phillip and his team for organizing this very exciting conference and course here in New York City. So I will be talking today about protein-protein interaction, and we've heard a lot yesterday about signaling pathways and should we target a single molecule or whole pathway. So we'll try to go a little bit deeper today into protein-protein interaction and try to present you where do we stand today, what have we done in that field, why are those targets important for drug discovery. So I would like to first start with the pathways of drug discovery. So when we start any kind of project, there's a time where we look for a target. There's a target validation stage, and we're also looking for hits and to identify hits. Then comes the phase where we have those small molecules as hits, and we want to refine them, bring them to a different level, something that can be advanced further in the pipeline. And this is a very difficult phase for all of us doing drug discovery. So basically, we're trying to catch that ball, but sometimes it's impossible. So we are there and we are stuck and we not always understand what's going on. But at the end of this phase, we reach the time where we have discovered a very interesting novel molecular entity, which now is going to be our selected clinical candidate. So we hit our target and we hit our goal in this case. So finally, after a regulatory approval, this chemical entity is marketed and it's time for us to relax and move on eventually with the next type of project we're working on. I don't want to comment too much on this slide here. This is really one way of representing the drug discovery process. What is important to understand here is that it's a very complex work. It's very difficult for us to take a biological target and find more compounds that will advance to clinical trials and become new drugs. Important also to understand is that this is a stage-by-stage quality assessment. In this analysis of the drugs which have been approved so far, there are approximately 20,000 drugs. Once one does a triage of those drugs and eliminates everything related to nutraceutical, different formulation, we are left with about 1,357 unique drugs. Out of those drugs are targeting 324 targets for all the different classes. So if we take this analysis and then expand it and to all proteins which have 35% identity to those 324 drugs, this number of approximately 1,000 drug targets came out. Those are the predictions today that we might have that many druggable targets. If we look a little bit more in this specific representation here, one can find out that G protein coupled receptors, nuclear hormone receptors, and ion channels represent more than 50% of the drug targets. So what have we done so far? Are those the low-hanging fruits? So one area which is very important for us these days are protein-protein interaction. Those are classified as challenging targets. So protein-protein interaction has been the holy grail of drug discovery in the pharmaceutical industry. However, they have been very difficult to tackle. It's been very challenging for us to find molecules interacting with those type of targets. We saw yesterday this slide, which is from a publication from Lipinski. So what we have here is basically the chemical space, which is almost infinite, and there is a very large number of possible small molecules with a molecular weight of 500 or less. In that space is not dispersed in such a way that we have now very specific islands for each of the different classes of drug targets. If we look in this slide a little bit closer, there is nothing about protein-protein interaction yet. So this is really an emerging class of targets, and then we look into that. So we have in this area the definition of druggability or druggable targets as defined by Lipinski, and also representation of the rule of fives. And we'll see throughout this presentation how we can apply those, or if they would be applicable to protein interactions. The drug platforms that we have today, what are they? So we basically have three major drug platforms. One of them is small molecules. The advantage is that those are orally bioavailable. They are cell and penetrate the blood-brain membranes. We also have peptides and biologics, which are covering antibodies and protein therapeutics. For peptides and biologics, we have a very high specificity, low toxicity, and they have the ability to block protein-protein interactions. So all of those have several disadvantages as shown here. And mainly for peptides and biologics, they are not cell penetrant. At least so far there's not been much progress in this area, and this represents an issue. So based on a publication of Greg Verdine at Harvard, and he came back to this number telling us that there's about 75 to 80% of all existing targets which are beyond reach of established drug platform. So he came to this number because we have approximately 30,000 genes. So out of those 30,000 genes, 3,000 of them are expected to have a small hydrophobic pockets which can be amenable to small molecule drug discovery. Another important feature of his analysis is that there's about less than 10% of those genes which are either secreted molecules or located on the cell membranes. Protein-protein interaction, I think most of you, or all of you in this room are very familiar with this type of targets. So as we know, there are crucial elements in cellular function, and human disease can be caused by aberrant protein-protein interactions. Also, there's been advance in understanding the mechanism of cell signaling, and since we know better about those different cell signaling pathways, we also have a better clue about where to interact or where to intervene around those pathways. So they're very important because protein-protein interactions may provide novel mechanisms to modulate different neural function downstream of the receptor activation. And as I said earlier, those are the high-hanging fruit. They're very high reward targets, but very high-risk targets at the same time. So what's the rationale for targeting protein-protein interaction in the CNS? So as we know, there's a host of cellular functions which require protein-protein interaction in the CNS. And also, as mentioned, those targets may provide novel mechanisms for us to modulate neuronal function. There's also mention in the literature that we might expect a higher specificity by targeting protein-protein interaction, since they might provide a measure of tissue specificity. What has been the strategies for neurodegenerative disease? One of them has been to inhibit, inhibit protein aggregation, and also the main one is to target the signals downstream of the signaling pathway. This slide is just a general summary about protein misfolding and and aggregation in neurodegeneration. And if we take a look at those 4 different diseases, such as Alzheimer's, Parkinson's, Huntington, and ALS, all of them have in common the aggregation of different type of proteins. And this table is showing what type of proteins are responsible of those aggregation and the location of those proteins. And everybody, I think, in this room is very familiar with this. So what's important to take out from this slide is that neurodegenerative disorders may share common targets for therapeutic development. So one opportunity for us is, for example, to interact with those different proteins here, and especially to try to block the formation of the oligomers in one way, or to block the production of those oligomers. But also an approach that people have used is to try to find agents, small molecules that will bind to those different proteins and then block their interaction with other proteins than themselves. The other area that people have looked into, and this is a very general slide which shows the G protein coupled receptors. So as we know, for G protein coupled receptors, we have an extracellular signal which is transduced through the cell membrane and then activate the heterotrimeric G protein, which led to a dissociation, and we have the G alpha subunit and the beta gamma subunits. So what people have tried to do now is not to find agents that will interact with the extracellular ligand and the receptor, but to look at different type of compound that will interact either with G alpha. Actually, there's been very little success in this area, or to interact with G beta and gamma. And there's been examples of G beta gamma inhibitors described in the literature. Another area of interest for this specific class has been to look at RGS proteins and try to find compound that will interact with RGS. And in here, there's a little bit more success too. So this slide was mainly to show to everybody that it is possible to look into molecules that will interact downstream of the receptors, and there's a lot of efforts in that area. So this is an area in which, for example, people have been very successful with kinase inhibitors, and we might hear about that a little bit later in this presentation. So when one is looking into interacting with targets which are within the cell membrane, there are significant hurdles now. So now we have to design compound that will be cell penetrant. And also we have to think about the pathway selectivity and any kind of issue which will arise with that. Protein modulation. So what is the challenge? So the major challenge from a drug discovery design is basically that we have a very large protein-protein contact surface and flat interface which are lacking any kind of suitable pockets. So if we think about that, we would say this is really undruggable. There's not much that we can do with those type of targets. There are also intrinsically unfolded proteins, so some type of proteins which have no conformation until they will interact with the ligand. There's been a low success rate in high throughput screening campaign, and fortunately, there's recent breakthrough in that field. One of them being that when we consider the protein-protein interaction surfaces, not all amino acids in these interactions are important for binding. And this led to the theory of the hotspots, which was first described by Jim Wells. And basically now this gives us opportunity to really design molecules for those specific hotspots, which has a surface area of approximately 600 angstrom square. So there's been also success story in small molecule inhibitor discovery for protein-protein interaction, and mostly in oncology. And we are waiting for this type of success story for CNS targets too. Are there any evidence of tractability for the protein interaction classes? So there is some here. So this is the the example of the BCL2 family of proteins, which are previously thought to be undruggable. So in this case, what happened, so very recently, this compound ABT737, which is shown here, has been discovered as a selective inhibitor of this protein-protein interaction. And clearly, this is an important breakthrough for people working in that field. If we look at those compounds from a drug discovery, from a drug design point of view, they're a little bit larger than what we typically see. They don't look very drug-like, but still those compounds, the first compound, ABT737, entered clinical trials, was not orally bioavailable, but then medicinal chemists were able to convert this compound to ABT263, which is now orally bioavailable, and also in different clinical trials for oncology. This compound was discovered through a fragment-based drug discovery. I just want to spend a few minutes here to describe this. So this is because we heard yesterday a little bit about fragment-based discovery. So this is very different from a conventional high-throughput screening where one would purchase compound collection from different vendors or have access to compound collection within an academic environment. So what we do here is very different. So fragments are typically very low molecular weight organic molecules, and we look at the potency between the 10 millimolar maybe to the 1 or 100 micromolar range. So we just want to find small pieces that bind to the protein and to understand, do we have a binding pocket here? Do we have something we can expand and later grow those fragments into the true small molecule that will become our lead and eventually enter our clinical trials? So protein-protein interaction, what are the mechanisms to modulate them? This is an example of two proteins. And one possible strategy is to look for hotspots, and those hotspots has been discovered. So binding of a small molecule to this hotspot will basically break the interaction between those two proteins, and now the protein complex is not forming anymore. The second strategy is to look for allosteric sites on those proteins. And in this case, binding of the small molecule to the allosteric sites will also lead to an allosteric changes in this protein, and now they cannot bind together anymore. So there's been a lot of progresses in computational methods in order to try to identify those hotspots or to identify potential allosteric sites or binding sites. The second approach for the mechanism of action of PPI modulator is basically to stabilize the protein complex. So in that case, we have a ligand which has been designed now to fit both a binding pocket in protein 1 and a binding pocket in protein 2. So that way now we stabilize this whole complex. So we heard yesterday about rapamycin, which is an agent which is acting a little bit in this way, through this type of mechanism by binding FKBP12, and then the rapamycin FKBP12 complex will sequester mTOR and then stabilize this whole complex. In this specific strategy, there's an important drawback, because now we want to design ligands that will really interact with both proteins. So typically those ligands are large, so large that they might not be as druggable and as interesting for what we want to do, and we'll look into that a little bit later. So what is the problem in protein-protein interactions? What are they so difficult for us to tackle? So as I said earlier, there's been a very limited success in high-throughput screening. As we know, protein-protein interactions are very different from the more established drug targets. So what we've done so far in the drug discovery industry, and I think Klaus Lipinski described it very well. So we basically said, let's have large collection of compound, let's have high throughput screening capability and screen any targets coming to us. So we applied it to protein interaction, and not surprisingly, we didn't find many very interesting hits through this specific approach. So what we learn actually is that the current small molecule compound collection are simply decorated in a wrong way. So as we can see here, we have different small molecules with different shapes. And as you can see, they have very different shapes. So they might not fit the protein-protein interaction pocket. So where is the biologically relevant chemical space of protein-protein interactions? We don't really know that yet, so we are still learning in these fields. And what we know for different publications is that the molecular weight for protein-protein inhibitors is greater than 500, and it is predicted to be in the 600 to the 750, to the 600 to the 7 Dalton for small molecule protein-protein modulators. The specific strategies which are used for to design a protein-protein modulators are shown here. So we heard about high-throughput screening. We can screen small molecules, and as long as we are very careful by our selection of those small molecules. And also natural products have been very successful, and we heard a very good presentation yesterday on natural products. And one popular approach has been to use peptides and peptide libraries like peptide phage libraries to discover those different hotspots and then to apply computational chemistry in order to do, for example, a virtual screening of our small molecules. We talked about the fragment-based drug discovery, and this is really becoming a very popular approach for protein-protein interaction. There's been more than 50 different compounds discovered and entering clinical trials, which emerge from a fragment-based drug discovery approaches. Finally, computational chemistry methods, as I said also earlier, a lot of progress in this field, and it's still a field which is at the early stages for protein-protein interactions. Just a few words about the project discovery flow and the key elements which are important here. So first, it's important to think about what compounds do we select to screen against any targets, and more importantly, what compound do we want to screen against a protein-protein interactions. The choice of the different assays are also very important. So typically in this type of strategy, we would start with a binding assay. So it's very critical when this primary binding assay will be followed up with a functional assay which will tell us that binding to the proteins really affect the function of this specific protein. Critical is also to understand the selectivity and the brain permeability of the compound. We went through that yesterday. And building a multidisciplinary team. So it's important that chemists and biologists really work together to successfully advance compounds up to a preclinical proof of concept and even beyond that. What are just a few words about innovative approach in the field of protein-protein interactions. In one extreme, we have small molecules as shown here, which are buried within a binding pocket. And we have antibody, which basically bind surface protrusion. Recently, Greg Verdine has developed, and others have developed staple peptides. So what they do, they basically linked amino acid side chains of peptides, and the effect of this is quite interesting. First, the peptide conformation is fixed in an alpha helix in an alpha helices. And also they have demonstrated that those peptides are now cell membrane penetrant, and they can hit those targets which are located within the cells. Other approaches in the same area, protein epitope mimics and miniature proteins. And finally, something very different is called the SynBody. This is a synthetic antibody which work in a very different mechanism than any other methods shown here. So basically, synthetic antibodies bind to the surface of proteins. So this is a very novel technology which is actually developed by SynBody biotechnology, and I will be happy to talk with you about it after this presentation. Finally, we're just as a summary, but also to give you future perspectives. So we've made significant progress in developing small molecules for protein-protein interaction, especially in oncology. As I said, there's a compound or the many compounds entering clinical trials. But despite those progresses, there is substantial challenges ahead of us in developing protein-protein modulators for CNS indication. So the drugability of protein-protein interaction targets really depends on advances in different technologies as shown here, and from both the biology and the chemistry side, including computational chemistry. It's very important that you really come up with novel methods, novel way to really increase our knowledge of protein-protein interaction targets and discover novel agent for this target class. And this is everything I had to present to you today. And thank you for your attention.
Some time for questions. I was curious, Gerard, if you could comment on whether there is a difference to approaches if you're targeting protein-protein interactions that are the self against the self protein. So if you're targeting an interaction of one protein with its monomer, or versus one protein with its another target protein. So in other words, for example, in A-beta or alpha-synuclein, where you have these self-aggregates, is there a different strategy to targeting those protein-protein interactions versus BCL2 VAX interaction?
Well, yeah, it's a very good question. I think at the stage we are at, it's very difficult to answer this question, very specifically. But typically, we're always looking for the same things. Are there very specific amino acids in those oligomers or in those smaller peptides or in those proteins? Are there any specific amino acids important for the binding interaction? So we really want to look and to find those. If we can predict them,